Good evening everyone. We'll continue with the chapter of biomolecules of class 12th chemistry. This is the second part. We'll continue from fructose onwards. Now fructose also has a similar molecular formula to glucose C6H12O6. The major difference being it and glucose is the difference in its functional group. Fructose contains a ketonic group at its second carbon position, as you can see in the given diagram. It also belongs to the D series. It is a levorotatory compound and it is written as D minus, as we have discussed before of the configuration part. The structure is given here. CH2OH at both the terminal ends. Second carbon has the ketonic group and then HOH-OH. It only exists in two cyclic forms which are obtained by the addition of OH at C5 to the carbonyl group. The ring thus formed is a 5-membered ring and is named furanose because it looks similar to the compound furan as given below. It is a cyclic compound with 1 oxygen and 4 carbon atoms. Here is the cyclic structure of fructofuranose in beta D forms and alpha D forms. In alpha D form, the OH negative ion on the OH group on the second carbon is present on the right, and is this compound, it is present on the left. Here is the cyclic ring form. Now the comparison between glucose and fructose can be asked as a part of your examination. So kindly remember these basic differences. Both have the same molecular formula. One is a polyhydroxyaldehyde and the other is a polyhydroxyketone. Glucose has a higher melting point as compared to fructose. Glucose is dextrorotatory, fructose is levorotatory. Tollens reagent gives you silver mirror in both of them. Failing solution gives both of their test. Molish test is given by both of them. Phenylhydrazine gives similar test, and oxidation by concentrated nitric acid in glucose gives you saccharic acid, and in fructose it gives you a mixture of different acids. Now disaccharides. The two monosaccharides are joined together by an oxide linkage formed by the loss of water molecule. Such a linkage between two monosaccharides unit through an oxygen atom is known as glycosidic linkage. Our first major disaccharide is sucrose. One of the most common disaccharide, sucrose, is formed on hydrolysis on equimolar mixture of beta D glucose and beta D fructose. These monosaccharides are held together by a glycosidic linkage between C1 of alpha glucose and C2 of beta fructose. The major thing which you need to remember is the type of linkage and what are the products based on hydrolysis. Structures are not important for your examination point of view. Sucrose is also dextrorotatory. Maltose is another disaccharide. Maltose is composed of two units of alpha D glucose only, in which C1 of glucose 1 links with C4 of glucose 2. This structure is not important for you. Just remember the type of linking and the hydrolysis product. Similarly for lactose, it is also known as milk sugar. It is also a disaccharide. It is composed of beta D galactose and beta D glucose. Fischer projection of both of them are given below. Here is beta D galactose for you. Now we can see that here that stereoisomers which differ in the configuration of only one carbon other than the anomeric carbon are called as epimers, and that carbon atom is called as epimeric carbon. Hence we can say that beta D glucose and beta D galactose are epimers of each other. The linkage between C1 and C4 of galactose and glucose is present in the form of disaccharide linkage. Again, the structures are not major for you. You only need to remember the structures of glucose and fructose. Now let's continue with polysaccharides. Polysaccharides contain a large number of monosaccharide units joined together by glycosidic linkage. They are mainly used as food storage. For example, starch, it is the main storage of polysaccharide in plants. It is a dietary source for humans also. It exists in two different polymers of alpha glucose, amylose and amylopectin. Their difference is asked a lot of times, so kindly remember. Amylose is water soluble and constitute around 20% of the starch. Chemically, amylose is long unbranched chain with 2000 alpha D glucose units held by C1-C4 linkage. Amylopectin, on the other hand, is insoluble in water and constitutes about 80% of the starch component. It is a branch polymer and also undergoes C1 and C4 linkage. The structures are not important for you examination. Now cellulose. Cellulose occurs in plants in excess and is one of the most abundant organic substances to exist on our earth. It is predominantly present in the cell wall of different plant cells. The structure is not important for you. It is formed by beta D glucose units of C1 and C4 carbons. Glycogen is similarly the carbohydrate which is stored in the body of animals as form of starch. It is chemically structurally similar to amylopectin also, but it is more highly branched. It is present in the human liver, muscles, brain, etc. Whenever your body needs glucose, enzymes break the glycogen down into glucose. Glycogen is also found in yeast and fungi. Here is a summary table for all the types of carbohydrates, their types, reducing or non-reducing, their units and their type of linkage if any present in them. You can pause to revise the content or make a table of your own. It would be better for memorization purposes. Now a short note, all the carbohydrates which contain the CHO group or alpha hydroxyketonic groups or hemiacetal groups are reducing sugars. All reducing sugars show the phenomena of mutual rotation which we have discussed in part 1. Now proteins, the second part of the chapter. We have completed the major portion from where most of the questions are asked. Now we will contribute to the minor portions of the chapter. Now proteins. The word proteins is derived from the Greek word proteos, which means primary or of prime importance. All proteins are polymers of alpha amino acids. Now what are amino acids? Amino acids contain amino, NH2 and carboxyl C double bond OH. Functional group present depending upon their position. Amino groups with respect to carboxyl group. Amino acids can be classified as alpha, beta, gamma, delta and so on. Only alpha amino acids are obtained on hydrolysis of proteins. They may contain other functional group also. All the different amino acids have trivial names which usually reflect the property of that compound or from its source. For example, glycine is named since it has a sweet taste. In Greek, glykos means sweet. And tyrosine was first obtained from cheese. In Greek, tyros means cheese. Now let's discuss about the nature of amino acids. Now the general structure of amino acids can be given as here. A carbon molecule which has NH2 group, carboxyl group, hydrogen and an alkyl group. This alkyl chain decides what is the nature and which type of amino acid we are dealing about. Now these are your list of amino acids. There are total 20 in number. You don't need to memorize the structures. Just learn to differentiate between their essential and non-essential ones. The names of amino acids are given by the first three letter symbols. For example, glycine is given by GLY and the single letter code is G. Alanine is given by ALA and the single letter code is A. It would be better if you can remember the structure of glycine, alanine, valine. They can be possibly asked in the examination, but their probability is extremely low. Now the N, B and A which are present here are simply denoting the nature of these acids, whether they are neutral, basic, acidic. It depends on the number of amino group or carboxylic group, which one is higher in number. If there are more amino groups, then it is basic in nature. If there are more carboxylic group, it is acidic in nature. And if they both are equal in number, they are neutral, as you can see in the first 5 amino acids. Now all the essential amino acids in the given table has been marked with a star. These acids need to be obtained through a diet and our body cannot produce on its own, such as histidine, tryptophan, phenylalanine, methionine, threonine, lysine, arginine, isoleucine, leucine, and valine. These are essential amino acids which need to be obtained through our diets and cannot be produced within our body. Now, classification of amino acids can be classified as acidic, basic, or neutral depending on the number of amino and carboxylic group as discussed above. And the amino acid can be synthesized in our body are known as non-essential ones. On the other hand, which cannot be synthesized within our body are known as essential one, and they are to be intaken through our diets. Now, properties of amino acids are here as follows. They are colorless, generally crystalline solids. They are water soluble with high melting point and behave like salts rather than simple amines or carboxylic acids. This behavior is due to the presence of both acidic and basic components in the molecule. In aqueous solution, the carboxylic ion can lose a proton and amino group can accept a proton, giving rise to a dipolar ion which is known as a Zwitter ion. This is another part important for your examination point of view. Its definition and structure can be asked in the examination. So kindly remember the definition and the structure as follows. This is neutral but contains both positive and negative charges. In Zwitter ionic form, amino acid shows amphoteric behavior, which means they can act both as an acid and a base. Now this is an important exception. Except glycine, all other naturally occurring alpha amino acids are optically active. Glycine is the only one optically inactive because in case of alkyl group, the R chain in glycine is simply hydrogen only. Now let's continue with the structure of proteins. Now proteins are polymers of alpha amino acids as we have discussed above, and they are connected to each other by peptide bond or peptide linkage. Chemically, peptide linkage is a type of an amide which is formed when COH and NH2 group meet each other and the removal of water molecule take place. Carboxylic acid loses its OH group and NH2 loses one of its hydrogen to form CONH bond, which is known as peptide linkage. The reaction between two molecules of similar or different amino acid proceed through the combination of amino group of one molecule with the carboxylic group of the another. This leads to the elimination of water. Now classification of proteins. Proteins can be classified into two types on the basis of their shape. Fibrous proteins, when polypeptide chains run parallel and are held together by hydrogen and disulfide bonds, then fiber-like structure is formed. Such proteins are generally insoluble in water. Some common examples are keratin present in hair, wool and silk, and myosin present in muscles. Next, globular proteins. This structure results in the chain of polypeptides coil around to give a spherical shape. They are usually soluble in water. Insulin and albumin are the common examples of globular protein. Now the protein structure and shape is studied at four different levels: primary, secondary, tertiary and quaternary. Primary structure can be one or more polypeptide chains in a straight chain linking manner. These are a sequence of amino acids and their primary structure known as proteins. Any change in this primary structure can lead to a change in the sequence of amino acids. Secondary structure of protein refers to the shape in which a long polypeptide chain can exist. They can be exist in two different structures, alpha helix and beta pleated sheet. These structures arise due to the regular folding of the backbone of polypeptide chain due to the hydrogen bonding between carbonyl group and NH group on the peptide bond. Alpha helix is one of the most common ways in which a polypeptide chain forms all possible hydrogen bonds by twisting into a right-handed screw helix with the NH group on each amino acid. Tertiary structure of proteins represent overall folding of the polypeptide chain That is further folding of the secondary structure. It gives rise to two major molecular shapes, fibrous and globular protein, as we have discussed before. The main force stabilizing these are hydrogen bonds, disulfide bonds, van der Waals forces and electrostatic forces of attraction. Quaternary structures. Some of the proteins are composed of two or more polypeptide chains referred to as subunits. Subunits with respect to each other is known as quaternary structure. Denaturation of proteins. Proteins found in a biological system with a three unique dimensional structure and biological activity is known as native protein. When a native protein is subjected to physical change like changes in temperature or change in pH, the hydrogen bonds are disturbed. This leads to the unfolding of the helix and uncoiling of the protein. This is known as denaturation. The coagulation of egg whites on boiling is the most common example of denaturation in our day-to-day life. Now let's continue with the DNA part or nucleic acid parts. Now nucleic acids are mainly of two types, deoxyribonucleic acid and ribonucleic acid, that is DNA and RNA. Since nucleic acid are long chain polymers of nucleotides, they are also known as polynucleotides. Now chemical composition of a nucleic acid. On complete hydrolysis of DNA or RNA, it yields a pentose sugar, a phosphoric acid, and a nitrogen containing heterocyclic compound, which is also known as a nitrogenous base. In DNA molecule, the sugar moiety is beta D deoxyribose, whereas in RNA molecule it is beta D ribose. DNA contains four bases: adenine, guanine, cytosine and thymine. RNA also contains four bases. The first three are same and DNA, but the fourth one is uracil, which replaces thymine. The structures are not important for your examination point of view. You just need to remember the names of the bases. Now the structure of nucleic acids. One unit of a nucleic acid is formed when one prime position of sugar is known as nucleoside. When multiple nucleosides attach each other in the presence of phosphate group also, it gets converted into a nucleotide. This is a phosphate group which is known as PO4 3-. It gets attached to the nucleoside to form a nucleotide. Nucleotides are joined together by a phosphodiester linkage between 5' and 3' carbon atoms of the pentose sugar. You can see a simplified version of the linkage given here. The information regarding the sequence of nucleotides and chain is known as primary structure. James Watson and Francis Crick were the first ones to give a proper structure of the double strand helix of the DNA molecule. Two strands are complementary to each other in DNA and they are bonded together by hydrogen atoms, form specific pairs. Adenine forms bonds with thymine and cytosine forms its bonds with guanine. In secondary structure of RNA, helices are present which are only single stranded and they can sometimes fold back onto one another. These are the major type of RNAs as you can see here: messenger RNA also known as mRNA, ribosomal RNA also known as rRNA, and transfer RNA also known as tRNA. Now the difference between DNA and RNA as given here in the given table. You need to remember only 3 to 4 differences for your examination point of view. You generally get a 2 marker to write the differences between DNA and RNA. You can use the chemical properties or their structures. It is up to you. The biological function of nucleic acids. DNA is the chemical basis of heredity, that is the transfer of genetic information from one generation to another, and also serves as the reserve of genetic information. DNA is exclusively responsible for maintaining identity of different species over millions of years. And for the use of RNA, it is used for protein synthesis within the cell and for the synthesis of a particular protein present within the DNA. Now the next part is enzymes. Enzymes are biological catalysts. Chemically, all enzymes are globular proteins. Some are given here, such as invertase, maltase, lactase, etc. pepsin, trypsin, nuclease, DNA polymerase, RNA polymerase. You don't need to go into that detail. Just remember the basic definition of enzymes and a few examples along with it. Now moving on to the next part of vitamins. These are certain vitamins and their deficiency can cause diseases also. Their sources are given here. Vitamin A can be obtained from milk, cod liver, butter, carrots, and its deficiency can cause night blindness or xerophthalmia, hardening of the cornea of the eyes, and cirrhosis. Vitamin B1, also known as thiamine, can be obtained from the given sources, and its deficiency causes beriberi and loss of appetite. Vitamin B2, its deficiency causes inflammation of the tongue and cheilosis. Vitamin B6, its deficiency causes convulsions and dermatitis. Vitamin B12 or cyanocobalamin, its deficiency can cause inflammation of the tongue and mouth and pernicious anemia. Vitamin C, ascorbic acid, can be used through citrus fruits and its deficiency causes scurvy, brittleness of the bone and bleeding gums. Vitamin D, its deficiency causes rickets and control of the calcium and potassium metabolism. Vitamin E, its deficiency causes sterility, loss of sexual power and reproduction. Vitamin K, phytoquinones, they cause hemorrhage and blood clotting issues. Vitamin H and coenzyme Q are not part of your syllabus. Now the last part of this chapter is hormones. Hormones are biomolecules which are produced in the ductless endocrine glands and are carried to different parts of the body by the bloodstream where they control various metabolic processes. These are required in minute quantities and unlike fats and carbohydrates, these are not produced in the body. These are not stored in the body, but they are continuously produced. This concludes our chapter. Thank you for watching. We'll meet you with the final chapter of organic chemistry left, haloalkanes and haloarenes.